I do apologize, everybody. It's been a morning already trying to get here. All right, so thank you for joining me. I was very excited to do this Grand Rounds because I think that, we have a challenge in the residency and education of our student doctors for the future, and that is, there's going to be more and more of a presence of data that is really in the field of precision medicine. And so I wanted to try and give a beginning of that topic in using the field of autism as an example. And the title of my talk is Determinants of CNS Wiring in the Brain Implications for Cognitive Neuroscience. So really, the challenge here is for us to understand from a clinical standpoint how on the left side we can take genetic data, understand how the brain not only builds synapses but also builds circuits within the brain to affect the developmental trajectory of behavior, which is modeled by the different colored lines on the right side. Now, in trying to understand CNS wiring, I'm going to be talking about autism spectrum disorder. And just to remind everybody what autism is, it's the most common pediatric neurobiologic disorder, 2% of the pediatric population. And there is impairments in communication, function, impairments in restricted interest and repetitive behaviors, three issues. Can somebody turn off their mic?
He was born in 1967.
Can you turn off your mic, please?
He was born the day before you in 1967. She always made sure to wish you happy birthday.
Can you turn off your mic, please? Thank you. All right, and so the building of neurocircuits in the brain really goes through a number of different biological processes that is labeled here in the upper left part of this figure. Both the rodent and the human starts with neurogenesis and leading to neuronal migration and the building of the cortical subplate. And then you have. Can I ask you to please turn off your mics? Thank you. Anyways, so then building of the brain continues through birth and into the toddlerhood period with the building of synapses, apoptosis of cells that we don't need, and then, of course, myelination. On this screen, just to familiarize everybody with, the genetics of autism. Autism is thought to be about 80% genetic and about 20% environmental. We probably in many ways could extend that to other developmental disorders as well in pediatric neurology. The things that we can measure, of course, in the clinic are copy number variants, mutations, and single genes, and of course sequence variants that we can look at in exomes. But in total, our hit rate is probably no more than 50%, 40 to 50%. So 60% of the genetic mechanisms in autism and probably in many other different diseases are probably the effect of common genetic variants of low effect size, and thus all those together become a polygenic impact. The database for autism genetics has about 1,000 suspected genes that have come out of the autism genetic studies. At least 100 of them seem to be relatively strong susceptibility genes. Now, you notice I don't say causative genes because there really are, quote unquote, no autism specific genes, so to speak. There are more genes that they may cause autism, but they may cause epilepsy or schizophrenia or bipolar disorder. And a large majority of these genes in the database either impact chromatin, impact axonal guidance, or impact synapses, which is a common theme that we'll talk about during the lecture. So the pathogenesis of ASD on the left side, certainly as we're building the developing human brain, there is this three different areas: the marginal zone, the building cortical plate, and then the subplate. And normally the activity is synchronized, as in D, and it produces a normal cortex. But if the activity of the neurons that are in the cortical plate and the subplate either become asynchronous or have reduced activity due to a wide variety of etiologies, and some of which are noted there, and which can be environmental causes of autism, such as in utero infection, inflammation, drug exposure, hypoxia, but this also extends to the genetic causes as well. Can lead to persistent neurons, alterations of neurons, and cause cortical miswiring. The very interesting thing about autism is, and many of these neurological diseases that we see in the child neurology clinic, is it's really disease of GABAergic circuitry. And what is shown on the figure on the right side is that in A, there's overall synaptic plasticity that occurs through the time of birth or through childhood. And we'll talk about this. We'll talk about this cortical plasticity, and it leads to so-called critical periods. And those are periods in which neural circuits are modified, and the structure and the function are sculpted by experience. And if you've ever wondered the mechanism by which our therapies work, and of course in autism, behavioral interventions are the main therapy. It really works through modification of synaptic plasticity during these critical periods, and we'll talk about that more in the lecture here. And not advancing. Okay. So the pathogenesis of autism, besides altered cell density and function, also involves looking at really bigger brains. These brains probably include increased numbers of neurons within the frontal and the temporal region, with so-called bigger mini columns. Mini columns are essentially cell columns that go from layers 1 through 6, and function as kind of as a single firing unit to mediate the cortical function of that particular neural circuit. So as a result of all of these things, on MRI, you can actually measure increased curvature and cell surface area. And then on the right side, what we're trying to show here, this is a graph of the critical periods in synaptic plasticity of the various brain areas: somatosensory cortex, auditory cortex, visual cortex. over the lifetime of a mouse from P10 to P28, or roughly the neonatal period in humans, all the way out to 1 year of life. And of course, the green line extends out past the 1 year of life. And the idea here is, again, to show that particular neural circuits have definite critical periods in which cortical plasticity can be modified by experience and by therapy. And I just wanted to re-emphasize that point. So really, when we talk about autism is a disease of so-called functional connectivity, meaning that long-range circuits or between brain regions those circuits are functionally deficient, and within brain regions they actually might be hyperconnected. As a result, you might develop savant abilities. For instance, from the film Rain Man, if you recall, he had an incredible ability to calculate numbers, and that is thought to be due to the increased synaptogenesis within the angular gyrus. This particular slide is to remind us that really cortical networks really come in three different flavors. There are these low-level local networks that are either basic motor or sensory processes, in which the circuits are highly segregated and they basically don't have a lot of interacting parts. And then there is these higher, in the second to the left there's these higher level distributed circuits, which are the typical sort of circuits that we talk about in measuring in neuropsychology, which include working memory, language, semantics, emotion, social cognition, and attention. And the idea is that you have within network integration as well as local integration. And then finally, the brain has these transient meta-networks. So essentially, all of the different high-level distributed networks working together as a single so-called meta-circuits to mediate complex goal-directed and flexible behaviors and cognition. And as you think about autism, autism has defects both in these higher level distributed networks that we measure via neuropsychological tests, but they also have these defects in these transient meta networks as well. For instance, flexible behaviors. As everybody who's seen an autistic patient may know, their cognitive flexibility is not good at all. And that's a dysfunction of these transient meta-networks. This is just to remind us that there are, and I've got several circuit diagrams here, and I think the point of these circuit diagrams is just to remind us that, there is, the circuits that are involved in autism are basically circuits that are mediated by the frontal cortex, the parietal cortex, and the temporal gyrus. Here is the circuit diagram for so-called theory of mind circuit, that is mentalizing or understanding the basic thoughts and behaviors of others. This is the circuit for language, which has not only the participation of the frontal cortex and temporal cortex, but there's also involvement of both parietal regions as well as subcortical regions. And another really interesting thing about autism is that, both the right and the left hemispheres are not wired correctly between the hemispheres. In fact, there's a relative lack of hemispheric specialization. Over the last couple of years, there's been identification of so-called hemispheric specialization genes. And in autism, there's a relative shift of left hemispheric function over to the right hemisphere. And sometimes when you actually do neuropsychological testing and functional MRI, you can actually see this, as we saw with a patient with autism and epilepsy in the last couple of weeks in surgery conference. So, to summarize my introductory slides, normal brain formation is dependent upon activity dependent as well as activity independent processes, and particularly in reference to both autism and epilepsy, this circuit synchronization and critical periods are dependent upon GABAergic cell function. That cell function has a dependence upon energy, has a dependence on circadian rhythms, and that's part of the reason why sleep is a very important component clinically in both autism and epilepsy. It's transcriptionally dependent, and that's why some of the medications that our patients take during pregnancy, for instance, or the mothers take during pregnancy, for instance, valproic acid, can lead to autism, and it's also experience dependent as well. Normal brains definitely have right and left hemispheric specialization, and this is abnormal in autism. And just to remind everybody for the discussions later, neurocircuits come in these three different types: these low-level motor sensory circuits, these secondary level high distributed networks, and then these transient metal networks that mediate complex, directed, flexible, and behaviors and cognition. Let's now talk a little bit about some of the etiologies of autism. Certainly, we've talked about how autism is genetic in nature, and that certainly mediates the building of synapses and the building of the deficient circuits in autism, which leads to deficient trajectories over on the right side. But there's also an impact of the environment and so-called extra genetic causes in autism, and we're going to briefly talk about that. One of the very interesting things about autism is that there are these new emerging pathologies that basically go back to our understanding of what's happening in the brain cortex. And one of the very interesting things is that it's been seen in the last couple of years that neurovascular signaling, specifically signaling in the cerebral endothelial cells, may actually control GABAergic neurogenesis and differentiation. Now, one that's really very interesting is from two different standpoints. One is a story that comes from metals biology. There are a lot. Recently, there was an announcement that there's an increased concentration of lots of heavy metals, cadmium, arsenic, lead, other things like that, in baby food, far beyond what you would expect you would want in drinking water. So the impact of these metals on cerebrovascular endothelial cells is, can impair the blood-brain barrier, among other things. And that may lead to, as it shows 2 and C, a change in vascular signaling to the developing neurons that will change differentiation. Then another thing that has been noted of late is that, and I'll show you some of the data for this, is if you have genetic alterations in vascular genes, and I'll show you some data from pathogenic copy number variants from patients with autism or autism and epilepsy, you can actually show that those are associated with CNS symptoms in autism. And then in animal models, you can actually show that as a result of the breakdown of this developmental neurovascular signaling, that there's actually impact on the blood-brain barrier. That's a new emerging field, and we'll talk about that just very briefly, and then we'll go on to some of our work from the Big Data Project. I think this is fitting on most of the slide. This is a table that's from one of our posters, and that we're giving from at the International Society for Autism Research in May. And what this table shows is that on the left, it shows behaviors that were actually quantitative from questionnaires given to patients' parents. And what we're trying to show here is that these behaviors, which can be mediated by particular neurocircuits, are actually associated with genetic variants that are expressed in those particular circuits. So if you take the patients who have genetic variants expressed in those particular circuits, you compare them to patients who have a normal set of genes, what you find is that there's a statistically significant difference in some of these questionnaires that quantitate self-stimulatory behavior, anxiety, adaptive skills / daily livings, and motor function. And what was really interesting about all of this, so we've quantitated behaviors to circuits to genes. And what's really interesting about these particular genes that we identified that were expressed in these neurocircuits mediating these behaviors was that a fair number of them actually were vascular genes. And that led us to say, well, if that's true, then what about neurovascular signaling in mice? Well, it turns out that we've been studying neurovascular signaling in mice now for almost 15 years in a family of axon guidance genes, which, of course, axon guidance genes are important genetic causes of autism. They're a family of guidance cues of greater than 20 genes that are expressed in a lot of different places in the brain, as well as in the heart, lungs, immune system, in the kidneys, and importantly in cerebral endothelial cells. These genes, which are expressed in both interneurons and pyramidal cells, regulate neurogenesis, they regulate neurite outgrowth, they regulate synaptogenesis, or it regulates cell migration, and they regulate the number and the function of interneurons. And one of the particular genes that we study in the lab is the semaphorin 3F and neuropilin 2 signaling system. Neuropilin 2 is the receptor, which is an autism susceptibility gene. It is activated by this ligand called semaphorin 3F. And what was really interesting is that we had shown all of these different autism and epilepsy characteristics of this mouse model previously. And because of some of this vascular data that we'd gotten from humans, we started to look at the impacts of deficient symmetry of signaling on the blood-brain barrier. Um, and shown here is the stainings of the knockout mouse animal for two serum components, albumin and, of course, serotonin, that is from platelets. And what was very interesting is that both in the amygdala, the cortex, and the hippocampus, and the knockout animals, we saw increased immunostaining of both albumin, and as you can actually see there, the albumin was actually uptaked in neurons and actually outlines neurons very nicely in the amygdala and in the cortex. And so there's, and through a lot of other work in staining, we were able to show that there is increased serum components in the brains of these mice that have autistic-like behavior. We are also able to show that there was increased oxidative stress as well. And what's interesting about that observation is that, the genetic causes of autism have been shown to have increased oxidative stress in interneurons in many of the mouse models. And, certain intervention experiments have suggested that interrupting the oxidative stress in the interneurons ameliorates the autism-like behavior. So the bottom line from this emerging pathology data is that, it's long been known that environmental causes of autism may impact neurocircuits and ASD. But what's really interesting now is these data from human genetic experiments, the metal experiments, and genetic manipulation of mice suggests the hypothesis that neurovascular biology may be a new frontier during development. And in fact, it may not only be for autism, but may be for a lot of other neurovascular disorders, where that the neurovascular signaling impacts GABAergic differentiation, and as a result, you have maladaptive formation of neurocircuits and expression of autism-like behaviors. Now, moving on to humans, one of the big projects that we've had over the last couple of years, and this is a project that is called the Big Data Project in Autism. It's part of the Big Data Medicine Project that is a collaboration between the School of Medicine and the Speed School. And the idea here is how can we take clinically available data from patients who have a particular disease, or in our case for neurology or developmentally abnormal patients, how can we actually use that to diagnose autism and then predict treatment responses? And the idea here is to use integration of imaging data, genomic data, and behavioral data to lead to a decision of autism or not, number one, and then number two, if we can we predict the local neurocircuits that are autistic, and as a result be able to predict the types of treatments that the patient may individually respond to. So as you know, the pathway to the diagnosis of autism is really a very subjective process that involves a parent suspecting that a patient may have autism. The pediatrician then suspects that. You schedule an evaluation, and this happens over the first 18 months or so. And then there's a series of testing that may not only include the pediatrician, but may include neurology evaluation, an early intervention provider eventually looking at that, and then eventually getting to the psychologist anywhere between 24 months to 60 months and getting to diagnosis. And once you get a diagnosis, you can get the patient into treatment with behavioral interventions. But this is a long, seductive process, and the access to diagnosticians is not is not terribly great, and there's oftentimes a long wait, although we're working on that here at Norton Children's. So, is there a better way to be able to diagnose autism? And that's really what we have set out to do with our MRI experiments. So the objective is to determine which sets of neurocircuitries may be relevant to the human clinical phenotype in autism, and how can it predict treatments. And the working hypothesis is that the neurocircuitry that we identify early in development is responsible for the expression of the human clinical ASD phenotypes. The data sets that we have been using have been from the National Database of Autism Research, which is an NIH-sponsored database in which behavioral data, genomic data, and MRI data are all deposited. We have about roughly somewhere around 1,200 subjects that's in this data set, for which we have MRI genomic data and behavioral data. The question is, can we find a correlation between our neuropathological findings and the MRIs? We spoke about the increased brain size, which may be mediated by the large number of mini columns in the cortex. So the idea here is, can we use MRI to detect the changes in the cortical surface of an autistic subject versus a control subject? And I'm running a little short on time here, so I'm gonna go through some of our data that we found from structural MRI. And nonetheless, we're looking at data not only from structural MRI, but also from functional MRI and diffusion tincture imaging, looking at connectivity. The database that we're using is a very well-characterized database. It's called the ABIDE database. We're looking at data across about 6 sites, and these are the database is collecting longitudinal data. The data that we'll be looking at will be somewhere in the 200 to 300 subject range, and in the age range of 8 to 17 years of age. Important in the MRI analysis is that we identify whether a patient is autistic or not only with a global brain diagnosis, but also add a local diagnosis through looking at the different Brodmann's areas. And basically, the idea of this is that you take the data from the MRI on the left, you do a deep learning network algorithm. This is machine learning, in which you have 2 steps. The data goes through the middle step there, which is really a training set. You train the artificial intelligence to recognize autism versus typically developing. And then it goes to the final step, which is the classifier itself, or the experimental data set, in which you get both a local diagnosis in the different Brodmann's regions, as well as a global diagnosis, autism or not. I'm gonna skip the feature pipeline, pipeline how we did that, just in the interest of time. One of the very interesting things about this was that the MRI features that we saw that correlated with diagnosis did correlate with the standard psychological instrument that's used with psychology, that's called the ADOS, or the Autism Diagnostic Observation Scale. And not surprisingly, features in the structural MRIs, including volume thickness, volume or thickness of the cortex, or fullness index, which is a measure of how much the curvature of the sulci, as well as surface area, correlated very highly with the diagnosis. Is not surprisingly, the data also correlated with the different brain regions that you would expect, including the parietal regions, occipital regions, frontal regions, and temporal regions. And find a nice correlation with the ADOS and with connections that are relevant to ASD circuits, which may include language circuits or other different circuits, and we'll look at that in just a second. A very interesting output of this is that on the left side is the typically developing brains, and on the right side, C, D, G, and H is the autistic brains. And what you can see there is that there's more red, more yellow. Overall, it's lighter, and you can see that you can actually give a very local diagnosis to particular brain regions. And those are markedly different from the typically developing brain. And skipping the fusion of both the anatomic and the functional MRI data, what's important to understand is that whether you can look at using a bunch of machine learning classifiers that's in the table on the right, and your diagnostic accuracy, your sensitivity and specificity are, especially in the random forest classifier, is getting towards the 80% range, which is, pretty doggone good. And that is, done purely with quantitative biology, and really involves no psychology instruments at all, from a scan that may take you 30 minutes. And as I said, the neural circuits that you find that are autistic, the anatomic correspondence that's on the right side of this slide include frontal regions. The cluster 4 is in the prefrontal cortex, and the TPJ is the temporal-parietal junction. Those are Broadband areas 39, 40, and 22. And as you can see, they map to a bunch of different neural circuits that are involved in autism. Those with reward learning and habit, which mediate restricted interests and repetitive behaviors, those that have defects in social and social skills, those that have defects in language. And what's interesting about all of this, those who work in memory, what's interesting about all of this is that, you can identify 4 or 5 different Broadband areas that are markedly autistic, and it will impact a wide variety of these cognitive skills, and as a result, you can develop this whole spectrum that is mediated with autism. So our summary of our results, and I think I'm probably going to end here. Our overall accuracy, sensitivity, and specificity is in the 80% range. Our positive and negative predictability is getting better with each data set that we have. What's interesting about the data of the 8 to 17-year-olds is that we identified similar affected regions in neurocircuits that were found in the high-risk infants that were later diagnosed with ASD. So the brain regions in the neurocircuits are probably the same. Our prelim results suggested that these gold standard measures of autism that the psychologists have are significantly associated with these key brain regions that were also identified in infants. We still have a lot of improvement that we need to do. Our sensitivity and specificity per brain region is still not what you really would want it to be. You'd really want it to be over 90%. We have been able to replicate, this was the data of one of our original students. We've been able to replicate this data in the laboratory. 3 more students are currently working on that. And they're looking at getting greater numbers of patients, hopefully to help us to define the autistic neurocircuitry levels a little better, and increase our sensitivity and specificity for this work. We also are looking at gene clustering as well, but I'm not in behavioral mapping. The bottom line for both of that work is that the genes as well as the behavioral clustering data do map to these frontal, temporal, and parietal circuits that were identified in the diagnostic work. So the idea here is that the integration of the big data is really for us to take clinically relevant available measures and to use that to delineate specific groups of genes in a single subject affected, trying to map those genes in the affected autistic circuits to predict the behaviors of the patient. Then ultimately what we want to do is we want to be able to, as we develop this data, to track how patients respond to treatments and hopefully understand better personalized treatments for these patients. And that's what's essentially seen in this data. So thank you for listening to my seminar. I appreciate that. I apologize for being late. As I said, a lot of this work is in collaboration with a lot of different departments and schools, including the U of L Bioengineering, U of L Computer Engineering, the Pediatric Research Institute, and then, of course, our funding sources. Thank you very much. I'm happy to take any questions if anybody has any questions. And I apologize for not ending at 10 till. Well, is everybody still there?
Yes, we are here.
I put everybody to sleep.
No.
Greg, yeah, can I ask a first question?
Sure.
You mentioned a little bit about the mitral impact probably for the autism behavior or disorder. And from the human state, is there any epidemiology study shown that is really related to some higher metal or trace element homeostasis, not just really higher metal or change the trace element like zinc, copper, ions, a ratio, something like that?
Well, there certainly are genetic data that speak to that. For instance, data zinc transporters and metallothionein genes. So there are some genetic data that relate to that, but certainly there has not been a systematic investigation of so-called genes-environment interactions in autism yet, and certainly that is some of the new funding from the Environmental Health Institute is looking at that currently.
So you agree from clinical endpoints, this is one direction we need to work on, right?
Yeah, I think so. I mean,
Good.
the data that is coming out, even on the anticonvulsant world, where the pregnant women are at higher risk for autism, suggests, as well as the data from immune signaling work in non-human primates, suggests that we need to have a better and greater delineation of the non-genetic causes / environmental causes of autism. And it may very well be that the variability that we see in patients may be due to the impact of the environment in every single patient, and that's likely to be the case. So understanding that and how to minimize those risks, and because they're preventable, is an important thing to understand.
Yeah.
Hi,
Any
Greg.
other question?
Yes, here, Jinkai. I have a question. Is there any clinical support, the link between the mild neurotrauma at early life stage and the autism?
So early life trauma and autism. Well, certainly, we know that mothers who have stress during pregnancy, particularly traumatic stress, they're at increased risk of having infants that eventually are diagnosed with autism. That is certainly true also if you do maternal stress in mice as well. The offspring are more likely to have autistic-like mouse behavior, for what it's worth. So there is some data there. I think that early life stress is yet another area that we need to understand in autism. For instance, preterm infants are at increased risk of autism. It's pretty stressful being in the NICU. So the question is, what are those? Is the stress in those preterm infants is that also leading to predisposition as well? So there are lots of different stress-related questions in pregnancy and certainly in the NICU that we need to answer related to the etiology of autism that's not necessarily genetic in nature.
Thank you.
Great. Any other questions?
Dr. Barnes, it was a nice presentation. A question.
All right. Yes.
Is there a gene panel which is being developed for these vascular genes which have been involved in the fetal neurobiology? Because that would be interesting to see.
Yeah, no, that's an interesting question, and it is relatively a new concept over the last year or two. Now, some of those genes are on the gene panels, but a vast majority are not. And so, that certainly is something that we probably ought to be talking to the genetic companies about, because it's certainly when you look at cerebral endothelial cells, they're much different from endothelial cells throughout the body, and they have GABAergic signaling, they have glutamatergic receptor signaling, so they have many of the signaling systems that are found in neurons themselves. So there's a lot more to be learned there, and certainly, as we learn more, we need to be able to convince the genetic companies to put those on the gene panels. All right, thank you. I think it's after 9 o'clock, so we'll end there. Thank you for the questions. Everybody have a good day. Bye-bye.
Thank you.
Thank you. Bye.